Okay, my wonderful friends, once again I intend to show that viruses are another thing that are not understood by science yet. They're just starting to dig into viruses. Now, I've been studying these for quite some time now, and I'm going to tell you right now, a virus is nothing more than an enzyme. We like enzymes, they work for us. Viruses work against us. Well, they think all viruses work against us. No, they don't. And let me just play this. This is from Seeker. And this just came out just shortly ago, and it's only a few minutes long, and I'll link this to you. But scientists who searched the hidden corners of the world for viruses poised to become pathogens is what she's going to say. And this, again, is from Seeker, and they're very good, an excellent channel to subscribe to. Now, this is what she has to say, and it's excellent. They do a very, very nice job. Here goes.
Become human pathogens. In many cases, they're searching for disease X. And that's not a name of an actual illness. It's what we call the hypothetical infection that could cause the next big global disease. The idea is to find it before it finds us.
Now, don't forget now, they are looking for viruses. I am going to explain to you precisely what a virus is, and precisely what an enzyme is, and precisely what a catalyst does, and precisely how they all relate to each other. But let's see what they have to say first, then I will do my preciseisms.
But other researchers are delving into what else viruses are doing on our planet besides making people sick. It's estimated that there are about this many individual viruses.
All right, there's this many individual viruses. Well, let me tell you something. Again, yes, I can go along with that because these are nothing more than chemistry sets. Every one of these is a package of chemistry. That's all it is. A virus and an enzyme, identical. They are program codes. I'm going to explain it to you in extremely great detail. I hope you'll be able to sit through it because this is a little complicated, but it's not all that complicated. They're identical. And now they're starting to understand this. They're looking into this, and they're finding all of these things they never expected were there.
On our planet. And we've only made the most minuscule dent in that number. We've found and classified about 9,000 of the viruses here
out of a quintillion, zillion, bazillion.
on Earth, and only about 200 of those are known to cause disease in humans.
Exactly. There's only 200 of them there. Out of all of these bazillion viruses. Well, they're not viruses. They're program codes. The viruses that hurt us, yes, they're program codes too. But those program codes go in and they say, you know, make this chemical, and that chemical hurts us. We should be able to have a program code in us out of that bazillion, zillion codes that says, hey, tell that thing to shut down. We don't want that happening here. That's not for us. That goes to the crabs and the mussels and the, you know, giant squids or something, you know. That's not for us. Get it out of here. So if we don't have those right program codes, we're going to take another bad program code comes in and takes us over. Not everybody gets the same sicknesses. Why? Some of them have the right codes to fight back against the codes that come in. These are codes. Those codes control bacteria. Now again, I'm going to get very deep into this. This is one of my passions in life now because I've been studying this for quite a few years, and I've known since, well, a long time that they're just missing how the human biome works. And even now, I can see, well, I don't know, we'll find out.
But over the past decade, scientists have been identifying new viruses by the thousands. And the main tool they're using to do that is metagenomics. This is actually something I'm using in my own research into bacterial communities, so let's break it down together. All
right, she's going to be going into genomics, basically, which is nothing more than sequences of these CTAGs, which are the protein codes that make up DNA. And if they're in certain, it's just exactly like a program. And that's all it is a program. Well, I'll show you this for another minute or so because she gets into a little more complicated than we care about. She's going to get into what they used to call serial programming, where you have to read one line of code continuously, and then there's parallel programs which they're, you know, like 1,000 codes a program are read at the same time, so you only have to read 8 characters and then you come out with your answer. The other one, you have to read 8,000 characters before you can get the answer. So it's faster, that's all.
Metagenomic sequencing, we take a sample, that can be soil, ocean water, a bodily fluid, and we purify it down to the genetic material of just the stuff we're looking for. And in this case, let's say it's viruses. So now we have the genomes of all the viruses in our sample, and let's picture each virus's genome as a piece of paper with sentences on it.
All right, now it gets a little bit complicated, but I understand this. I went to Johns Hopkins for genomics, and all it is it's just program codes, and I was in that business. I understand it extremely well. So a virus is a program code. An enzyme is a program code. They go out and they do a job. Now, let's dig a little deeper into this, because they're starting to understand there is bazillions of these, and why. And there's only like, they only have, what they say, a couple of hundred that are pathogens, or less than that, I can't remember now, but very few actually hurt us. And the ones that hurt us, if we have codes to get rid of them, just like I told you before, just get out of here. We don't want you here. We should have that code out of all those other codes. But if our biome is damaged and it cannot create the codes, because they are created from bacteria. And if we have antibiotics killing the bacteria that's in our gut, we don't have the programmers. I mean, the workers, the programs can come in, but if there's nobody to do the work, the bacteria is the one that has to create the chemistry. These are nothing, viruses and enzymes are nothing more than program codes, and they turn on bacteria that do the job. And they have also seen this just recently in new techniques of photography. Go call. Let me see if I can find that. Hold on. Okay, see, I told you, Seeker is the best. This is Go Call, I believe is his name, and he's at, I'm not sure what university he's at, but here's what happened. They changed the way they could see the light reflected back from these living creatures in real time. And before, it would kill and destroy the specimens, but they have a new reflective technology they're using where it's changed everything. So here's what happens. Here's Go Call, and he's going to reflect on the moment this thing happened. Now this goes back a couple of years ago, is when they first happened, and here it goes.
I still remember the moment. It's something I will never forget. The hair on my hands just stood up. It's a microscopic universe within each cell.
This is an unprecedented view of the cellular world.
Now, what I want you to take away from this, let's just start from the beginning. You see how intricate all of the tissues in your body are? This is the interstitium, and that surrounds all the cells and fills in all the spaces, and then you have these, like this is a white blood cell, basically. It's a leukocyte, I believe what they call them, and it has its bacteria living in this cell. Now, this is what cells actually look like. You see it up here? Now, this is, let me just start again. All right, he still remembers, and his hands...
Hair on my hands just stood up.
There you go.
It's a microscopic universe within each cell.
That's within each cell.
This is an unprecedented view of the cellular world, where we can actually see immune cells scooping up sugars in the ear of a zebrafish in real time.
Now, did you see what happened? Let's look at it back again, because this is just absolutely astounding. All right, this again, that's all it is a cell. And they're inside that cell.
World, where...
All right, here's what's going on here in my world. Somebody has alerted this immune cell to do some action, whether this is mucus or it's some type of a chemistry set, a catalyst, an enzyme, you know, a catalyst or an enzyme, which is nothing more than, in my world, a catalyst, an enzyme, a virus, I believe they're all the same thing. They are just a package of chemistry. And it's created whatever it needed, going around to harvest it from all these little blue guys. And it says, okay, take that out and go deliver it into the interstitial fluids, which is all through here, and it squirts it out. Now, this cell has to maintain its integrity from the rest of the system. It can't just be let anything in it come and go. It has to, this is an environment of its own. Once it squirts it into the interstitial fluids, that everybody plays a part in that. So it's a give and it's a take. What do we want to take in? Well, we want to take in things that we need to support this, and one of them is going to be the program code, and it's going to deliver that through one of the nerves, and the nerve somehow conducts that code down. I haven't really exactly figured it out, whether it's actually it's created somewhere and delivered, or whether it's a nervous impulse that tells the code to be create an enzyme. I don't know. There's a lot to look at, but here's what's actually going on in here. It creates whatever the product is, it squirts it out through the membrane. Just watch.
Where we can actually see immune cells scooping up sugars in the ear of a zebrafish in real time.
Focusing only on the crawling immune cells, we've noticed two classes of them. One.
All right, there's 3 classes of immune cells. Now, is what he's talking about. But they all have their own, he's talking about 3 classes of bacteria, really. And this type of bacteria is searching for problems. And then it reports to the other bacteria that creates the product. So here goes.
It seemed it was not hungry at all, but it was very active in terms of trying to figure out, what the environment is. There was another one that was kind of slouching around with a lot of food in its belly. We can actually conceptualize and visualize and analyze the contents of each of these cellular compartments in this crawling immune cell as it's kind of, scooping up its environment. I mean, that is a level of details no one's seen before.
We're living in a new era of cell biology, where
Yeah, they're starting to see things they've never seen before and start to understand the processes that are misunderstood, and they're still misunderstood because a lot of this experiments and this research just goes unnoticed, and it's rejected actually in most cases without examination. So it becomes a contest between who can find this and who can destroy the other guy's research. Basically, it's not, I don't see it as a cooperative effort to solve our problems. At this point, that's not what I've found. But Seeker is showing things, and that's all we can do is show things, because we cannot control academia. They seem to be a force unto themselves, and they seem to be in control of everybody's life at this point. Academia runs your life. You have to do what they tell you to do. You have to say what they tell you to say, or they'll destroy you. They'll fail you. They'll keep your money. And if you don't have a degree, you're not really in a good position. So you have to go to them, and then they take control of your life, and then they indenture you, and you're an indentured servant. And you dare not speak up against them, or you'll lose everything that you put into it. And the teachers, they're the worst ones. They're not allowed to speak. Not allowed to speak. If you do, you're done. Peer review makes sure that you'll be destroyed, and you not be able to speak once they decide you're not allowed to speak, and I'm not allowed to speak. So you're going to have to speak. You're going to have to share. You're going to have to talk to other people, and we're going to have to make all these things understood, because I'm telling you right now, probiotics will fix you. That's my feeling about probiotics. I've taken them. Everybody I know takes them is feeling much, much, much better, because they have the bacteria that can respond, and they have the protection that response affords. It causes mucus to be presented when there's under attack coming. These bacteria create other products that we're just not aware of yet. We haven't done the research. The research is totally lacking. I mean, they're seeing these things and, oh, look at that, look at that. Well, good. Let's find out where these viruses live, or these enzymes live, or these bacteria live. Where are they in the digestive system? They're all going to be different because there's all different types of digestion going on. Some are slower, some are quicker, some are up in your stomach area, some are in your throat, your saliva. They're all different enzymes that are in there, and those enzymes, they would call them viruses, but they are little chemistry sets that are sent out to say, hey, if you got some carrots coming in, here's how we deal with them. And the lettuce, oh yeah, you got to throw a little extra zinc in there for that, whatever it is. And I think the tonsils were one of the things that say, hey, you got some of this coming down, get ready. Or, you know, everything was a system, and it is a system in your body. Absolutely stunningly elegant interactions between cooperative organs and systems. It's just phenomenal. And the bacteria literally are 100% of the workers. 100%. Nothing happens without the bacteria working on it. And they keep things out of your good zone, and they bring things into your good zone. So they have to know what chemistry is not allowed and what chemistry is allowed, because the membranes are a two-way street. You can't just obviously you got to take in nutrients, and you got to get rid of waste. All right? You've got to fight off infections. You've got to fight off invaders. Mucus primarily does that, but then there's other things, the chemistry sets that can be sent out. And everything is a relationship between a program that says, here's how we deal with this thing. It's just like a manual. Here, hand them the manual. Oh yeah, okay, here's how we do it. And tell the guys to do it. And what if the guys aren't there because they're dead, they're bacteria guys? Well, not going to get done. Then you're invaded. And then, and I'm seeing this fibrosis now, this fibrotic condition everywhere. It's from acute respiratory distress syndrome, or even just pneumonia, or even just, you know, invasion of these collagens by an invading molecule that can't be destroyed by our system. And that's COVID, because we didn't know how to deal with this, and there's no program. So it comes in and it says, hey, I want you guys to do all this stuff. And they say, okay, no problem, we'll do it. You know, there's nobody telling us not to do it. Because normally you'd say, whoa, don't do that. Stop that. You guys are crazy, man. You're going to kill this guy. But we don't have those programs because we never saw this before. So, boom, the guy gets killed. And they have to keep forcing oxygen in because the springs won't open up in his lungs. It's an interstitium. They have springs loaded inside these fluid-filled bags, and they open up and close up, and open up and close up, and open up and close up. And then the same thing with the alveoli in your lungs, the bags. When the air goes in, they close back up. The air goes out, and but if it opens up, and air comes in, and it closes up, and it can't open back up again, you're done. Then they have to force air in. Intubation. All right, what she's going to explain right here is dysbiosis. It means that every system has to rely on every little bit and piece doing its job, and if you destroy one little bit and piece, it has a cascading effect and can allow for invasion and destruction of the entire system. So here's what she has to say. Here we go. The viruses in every there are viruses in every ecosystem. They help maintain our planet's biodiversity by keeping, here she goes.
It's biodiversity by keeping other species in check. So if viruses suddenly vanished from the face of the Earth, it's likely that the balance of other organisms would rapidly get out of whack.
Do you know what she just described? Antibiotics. She just described taking antibiotics. There's no difference whatsoever. You destroy your good bacteria, the other ones are going to take over, and they're going to invade you because the good bacteria is not there to do the job to, you know, like the plants and animals aren't there to do what they're supposed to do, so the biodiversity gets destroyed. And it's exactly the same thing in your gut, and then you're open for invasion. So she just described dysbiosis, or you know, a bad microbiome.
And one other major result of all this sequencing is a new understanding of just how thoroughly viruses have shaped life on Earth as we know it.
Just exactly like life in your gut. And they're starting to understand this A little bit. I mean, I'm not sure they're putting the pieces together right, though. I never see the pieces coming together right.
Because they invade living cells to replicate, they're equipped to take over a host's genetic replication machinery. And sometimes viral genetic material gets incorporated into the host. It's actually estimated that around...
All right, she just said that viral material gets incorporated into the host. Yes, absolutely it does. It gets incorporated in as a memory to how to respond to these viral attacks or how to make certain products your body needs. If your body says, you know, I'm in an area that has an excessive amount of something in the water, we need to deal with this. And it will literally, I believe, program and send codes into these areas in your DNA. They're called CPG islands in the junk DNA that are the store codes that are what we call read-only memories. They're ROMs, read-only memories. They say, go out and find what we do about this particular code, and it matches us up and it says, oh, here's the program that tells us how to deal with this toxic chemistry. And it might come back and say, create some mucus or create some zinc or copper or aluminum or, you know, this particular molecule and shoot it out into the, what they call the interstitial fluid system now. And there's a fluid system in your whole body that all the fluids in your body can mingle now, they found out. And so once you shoot this stuff out, it goes through your body and it's done. I mean, literally almost instantly. The body is just an elegant organism. Now listen to this now. It's estimated around 8% of human DNA actually came from a virus. Well, that 8% is these codes I'm talking about that did come from a virus. They implanted those codes in you to say, here's how we're going to deal with this, here's how we're going to deal with this, and they are the program codes. Those codes come down and then they go into the cells, and the bacteria take over from there. They say, oh, here's the code. Hey, everybody gather around here. Here's what we got to do. And then they do it. And I will show you that actually happening. Watch. And don't forget now, this is only 8% of your DNA, which is in your junk DNA. Because DNA, only 2% of your DNA, you know, is what they call good DNA, is what creates your structural, everybody's is the same. Only 2%. The rest, 98%, is your memory, your feelings, your understanding, your thought processes, you know, your emotions. It's all kinds of things are built in that other 98% of DNA. If only 2% is required to do everything elegantly that built you and us, I mean, just think of what the other 98% is capable of doing. And you're using 8% of it to memorize, well, they're saying it's now estimated. It could be, who knows what it is. Everything is an estimation, a guess. And all the guesses I've found are based on things that a lot of times just don't hold water. So we're going to see what holds water and what we used to call a sieve. Now, I'm going to show you these actual responses happening. I'm going to try to describe what the body is thinking about in my estimation. I could be wrong, but this, I would think it's worth a look. Now, I referred to junk DNA before, and I'm sure you've probably heard of it, but it's the science of no longer junk DNA. Now, when you're born, your DNA is 2% of it is used to do the functions of your body that they know about. Only 2%. And that's the construction of the body and all the functions and all that stuff. Now, so you got 98% they thought was just nonsense, just junk. And I'll show you that in a second. However, identical twins born that have identical DNA when they're born, exactly the same, they start to differentiate as they grow in life. And if they're separated early in life, they just completely, totally different DNA, because their life experiences are different. All their interactions, all their memories, all their infections, all their responses to their environments, totally different. And that is where that is stored in the 98%. That is my statement. Now, and I think they're coming along to understand this now. They're talking about, why would you study the junk DNA in kidney disease? Well, kidney disease, just like any other disease, if you don't have the program codes in there, then you can't respond. If you have the program codes and the bacteria is not there to carry out the instructions, you can't respond either. But here's the key. Among the 3 billion nucleotides in our genome, these are these little sequences, less than 2% are responsible for coding proteins. So 2% is what they think they understand. The rest of the sequences were long thought to be non-functional junk, possibly only there as a buffering zone for mutations so that we turn from slime into humans. Recent reports, especially those published by the ENCODE Consortium, who's really looking deep into it, indicate that by describing and annotating the cellular epigenomes, which are these sequences, functional roles could possibly be assigned to as much as 80% of the genome. All right, that's out of that 98%. Now, it seems that the instruction, listen to this is really important, listen carefully. It seems that the instruction manual that governs gene expression, which means what we always thought is the gene causes this. Well, it governs the gene expression, which is in the instruction manual, is hidden somewhere in that junk DNA. The programs are in there, but they're only programs. Listen carefully. The enzymes and the viruses are only programs. They instruct the bacteria what to do. If the programs are not there, the bacteria cannot do its job. If the bacteria are not there, the instructions can do all they want, but there's nobody to carry out the instructions. That is the key. So you have to have the codes, you have to be exposed to something a little bit, and then your body will figure it out and respond to it. But the rest of your insides have to be healthy so you can ward this thing off mostly. And then your body will say, hey, hurry up and fix this thing. Now, the people that have immunocompromised situations, they're already open to invasion because they've already got open places in their lungs and their nasal passages, and who knows, all the way down the digestive system, leaky gut they call it. Well, when that stuff can pass the barrier into you, it's attack time. Now, if you can't respond quickly enough, and this COVID is fast and nasty, and if you can't respond to it, it takes over, and its chemistry does its job and breaks down it's pretty. I understand what it does now. I've studied that pretty well. It causes fibritic issues where fibers break down in the springy parts of the blood vessels and the hearts and the lung and everything that goes like that. Those springs break, and then they get floating in this interstitial fluid. They collect in the deep vein thrombosis areas where they try to convert over back into from the arterial side to the vein side to bring it back up. And they clog in there, and then I did a video on it the other day. It was pretty deep, and it just keeps getting, day by day, I keep seeing more and more people are starting to understand this, and it's all become because people are starting to stand up for themselves and saying, we're not going to just accept what you tell us. We want you to look into this. And look into this and look into that, which has not happened, and they still are refusing. I've been 10 years trying to force a lot of this stuff into academia, and it has just been forced out, and it's still to this day. However, I can see that is changing. It seems to be really on a downhill roll for those people now. They have to come on or go get the hell out of the way. That's all. Okay, you would expect that being able to understand all of this different stuff is inside of a cell that would lead to some understanding, but it went just the opposite. It's just made things very confusing. Now, I believe this is an artist's rendition of what inside of a cell looks like, but it's based on observable sort of things that they can see and they just don't understand, but they can see these little structures and ribosomes and all these little carbon tubes and backbones and, you know, all kinds of things going on inside the cell. Well, how can we make any sense of this? What good is this? Well, I know for a fact that all of this different chemistry gives off a different spectral signature. And when you look at it, you can see all the different colors. Now, if you had a program that could understand all of this different sequencing colors and what they meant and, you know, what chemistry was in them, and you can, I'm sure they can do that. What I would recommend to do would be to take samples from a human body of all the different organ systems and the different areas in the organ system. Like the intestines, you have a small intestine, you have a large intestine, you have a colon, the bowel, you have all these different areas. And then along the whole line of it, it could be different, the bacteria, because once you get in there, things don't digest at the same rate of speed. So sometimes you may have to have a certain bacteria way down here because that stuff would have never got digested up here. And by the time it gets down here, it's ready to get broken down. So you need to take the whole system and find out what lives in every nook and cranny, and then be able to, a guy's sick or something, then be able to just get a little sample of that tissue or whatever and take a picture of it and see, you know, is this healthy tissue? Is what are we missing? What bacteria are not in there? What bacteria are excessive? What are, you know, that type of thing? What metals are missing? And you could, you know, with this new technology, and they could just know instantaneously with this artificial intelligence. It would scan everything and see the different colors and say, whoops, we got a problem over here. That green's not supposed to be here. Well, if it's green, it means it's copper. We're not supposed to have copper there. Well, why is it copper there? Well, we're missing a bacteria. And bacteria, there's a copper-producing bacteria. It's not there. Well, why isn't it there? Well, we got a breach in the, you know, whatever it is. And I don't think they understand the complexity and the way bacteria actually works to create all these different products that we need in our body. Because this is 100% driven by bacteria. They're the workers in there. They're like cities onto their own selves. And actually, I'm telling you, the way things look, they could be.